Today we're going to talk about functions of the nervous system and general nervous system subjects. But the first thing, the functions of the nervous system can be basically summed up in sensory input, that's gathered sensations, integration, which means interpreting and deciding what to do with that input, and then motor output, that's the reaction of an effector organ such as a muscle gland. The nervous system is divided into central nervous system, which is brain and spinal cord. And this is your integration and command center. The peripheral nervous system are your paired spinal nerves and cranial nerves that take messages from the central nervous system out to the rest of your body, as well as taking messages from the rest of your body back to the central nervous system. When we talk about sensory, we call this afferent. This is peripheral nervous system still. And afferent can be divided into somatic afferent fibers. This is coming from skin, skeletal muscle, and joints. And then visceral afferent fibers coming from your visceral organs. These are your hearts and parts and guts and everything that basically you find on the label of your salami. We also have motor or efferent, think of E for exit, division that transmits impulses from the central nervous system out to effector organs. Okay, the motor division of the peripheral nervous system is divided into somatic, which is voluntary. You consciously decide to control skeletal muscles, and then you send a signal and control them. And then autonomic, the word reminds you of automatic. You have no voluntarily control over this. Visceral motor nerve fibers. This is regulation of cardiac muscle, glands, smooth muscle, things that one can't control consciously. They run under the radar. You don't know what's going on consciously, and you can't control it. Okay, the two functions of these are your sympathetic, which is your fight or flight. When a bear attacks you or someone comes after you with a shotgun, your sympathetic nervous system will be activated. Heart rate goes up, bronchioles dilate, muscles go to your legs where you can outrun the bear or your classmates. Parasympathetic function, on the other hand, is when you're just sitting here chill, falling asleep during lecture, eating food, whatever it is you're doing right now. Okay, next we're going to talk about just some of the histology that makes up the nervous system. And our cells are divided into two principal types. The neurons, these are the actual nerve cells. They're excitable, they respond, and they do actually transmit electrical signals. And then we have our glial cells or neuroglia. These are supporting cells. The first of these we're going to talk about, this is in the central nervous system here, astrocytes. Abundant, very versatile, highly branched, and they cling neurons to synaptic endings, capillaries, support brace neurons. They also help determine capillary permeability, so they're involved in the blood-brain barrier. Growth, migration of young neurons, control the chemical environment, migration of, this has already been done, migration of young neurons, and possibly participate in information processing. That's kind of debatable. This is a picture of the astrocyte right here, and it's holding this capillary up against the neuron here, as you see. Now, another cell is a microglial cell, or microglia. Microglial is an adjective form. They are basically macrophages of the central nervous system. They eat things that need eating. Bacteria, fungus, viruses, but plaque, and things that are supposed to be inside your brain. Ependymal cells, basically squamous to columnar, possibly ciliated cells that line the central cavities of the brain, the spinal cord. They separate the central nervous system from the interstitial fluid, and in the cerebral spinal fluid in the cavities. I'm sorry, separate the central nervous system interstitial fluid from the cerebral spinal fluid, and they produce and circulate the cerebral spinal fluid. It fills the ventricles, the subarachnoid spaces, as well as the central canal of the spinal cord, as well as the subarachnoid space around the spinal cord. This is just a picture of them. Okay, in the brain we have oligodendrocytes, or in the central nervous system, and they create the myelin sheaths, and we'll be talking about these all through the lecture. A myelin sheath is a wrapper around the nerve cell. Now, in the central nervous system, the oligodendrocyte reminds me of a kind of, I don't know, a cyclops with multiple arms, or maybe an octopus. But it wraps around, one cell can wrap around multiple axons from different cells. That's different from in the peripheral nervous system. Okay, out in the peripheral nervous system, we have satellite cells that surround our cell bodies. Still kind of up for debate what they really do. And Schwann cells, also called neurolymnocytes, and these form the myelin sheaths around the peripheral nerve fibers. And also, if a peripheral nerve fiber, maybe the end of a axon, is damaged, they can help patch them back together. And here we have our satellite cells, as well as our Schwann cells. They're the little blue balloon looking things that wrap the axon individually. If you had a small enough group, they may be bundled. Okay, neurons are long-lived. They may live 100 years. Recently, there was a report that scientists think that 115 years is as long as you can live. I didn't know if your brain can live that long. They're amitotic. Once a brain cell is killed, it's dead. If you have a stroke and you can't walk, and then you learn to walk later, you have not repaired or replaced those brain cells. You have trained other ones to do the same thing, to teach you to walk. You have to learn over like a child. They have a high metabolic rate. They have to have a continuous supply of oxygen and glucose. And the plasma membrane in the neurons functions in electrical signaling and cell-to-cell interactions during development. And here we have again, this is in the peripheral nervous system. This is our cell body here. The nucleolus inside, this is the nucleus of the cell. These are your dendrites, they're receptors, and then they have one long axon that comes off of an axon hillock. And the little blue bubbles again are the Schwann cells wrapping around, making the myelin. And between these, you have nodes of Ranvier, which is the space where you see the yellow axon. Out here are your axon terminals, which secrete the neurotransmitters. In the central nervous system, we call a bundle of process a tract. In the peripheral nervous system, the same object is called a nerve. It's incorrect to call anything inside the brain or spinal cord a nerve. It's a tract. Okay, dendrites are your receivers. They tend to be shorter. They convey signals towards the cell body. The axon may be short if it's close to, like in your brain, close to another nerve cell it's trying to communicate with, or they may be very long. You could have one axon that goes from your big, from your spinal cord to your big toe, very long. They may branch off. We call these axon collaterals. They have, at the end of them, they have terminal branches, which are called teleodendra, and then knob-like axon terminals. And some people call them synaptic knobs. Some people call them boutons. Bouton is French for button. This is where you have the secretory region where neurotransmitter is released. The Functions of an axon is to conduct and transmit nervous impulses, which are also called action potentials, away from the cell body out to the effector. And movement of molecules. Since you have a very long cell, you have to move molecules up and down the cell body. Since things are made within the nucleus, such as mitochondria, membrane, they have to move towards the axon terminal, and we call this anterograde movement. We also have things have to go from the end of the cell, the terminal, up towards the cell body. That's called retrograde movement. Old organelles, but also viruses and bacterial toxins. And it depends on how long the axon is, on how long it takes for it to get there. And this brings up an interesting subject of rabies. This large, vicious dog here, raccoon and bats. Bats, raccoons, skunks, and wild carnivores are much more likely to give you rabies, but a dog could as well. But if it bites you on the toe, it may take you a year to have signs of rabies because it's such a long axon. If it bites you on the face, it's going to be a much quicker reaction. Either way, hopefully you would get treatment and not get rabies. Okay, the myelin sheath is the wrapper made that goes around the cells, either by the oligodendrocytes or by the Schwann cells, and it functions to insulate the axon and increase the speed of the nervous transmission. And in the peripheral nervous system, it's made by Schwann cells, and it wraps around in layers and forms a bulge called the neurolemma. Nodes of Ranvier are the gaps between the various Schwann cells, and this is the only place where axon collaterals are going to emerge. So this is showing wrapping round and round and round until it bulges, just like a balloon would bulge if you wrapped it around an object. And that is called the neurolemma. Some fibers are unmyelinated, and they may be wrapped in packages of 15 or more. So a typical large axon is going to be individually wrapped, and smaller ones may be wrapped in bundles. Now, in the myelin sheath in the central nervous system are formed by the oligodendrocytes, and one cell can form this basically processes off of them. So one cell can wrap many different neuron axons. They do have nodes of Ranvier. They do not have neurolemma, and some cells may be totally unmyelinated. And here's our oligodendrocyte again. Now, since myelin is white, white matter is made up of myelinated fibers, myelinated tracts in the central nervous system. In the peripheral nervous system, it's going to be myelinated nerves. Gray matter is nerve cell bodies. This is where the thinking and deciding place. Now, types of neurons, they may be multipolar. One axon, lots of dendrites. They may be bipolar. This is not typical. This is the retina is the main example I could think of. And then they may be unipolar, or actually pseudo-unipolar. Seems to be one branch, one process, but it branches off. Functional classifications of neurons, we have sensory or afferent. They transmit signals from sensory receptors towards the central nervous system. We have motor or efferent. They take impulses from the central nervous system out to effectors. Interneurons are also called association neurons, and they shuttle signals around in the central nervous system, and they make a decision what that nerve is going to do. And this is an overview of the peripheral, well, central and peripheral. And then it divides peripheral into somatic and motor. And then in the motor, it's divided into somatic motor neurons and autonomic nervous system, which is divided into sympathetic and parasympathetic. As we go along, you'll learn that both sympathetic and parasympathetic divisions are going simultaneously to maintain homeostasis. Now, nerves are highly irritable or responsive, and they create an action potential. And an action potential is an action potential. It doesn't matter what the stimulus was, whether it's a good stimulus, a bad stimulus. It's just a matter of where it's essentially wired in your brain as to what impulse you perceive. Now, electricity, you have to know a few things about this to understand action potentials. Opposites attract. Negative charge is attracted to positive charge. Takes energy to separate those charges. When you move the charges toward one another, energy is liberated. And if opposite charges are separated, you have potential energy. Voltage is a measure of the potential energy generated by a separate charge. Potential difference is voltage between 2 points, and current is the flow of electrical charge or ion between 2 points. Resistance is hindrance to flow of electrical charge. An insulator is something with high electrical resistance, such as myelin. A conductor is substance with low electrical resistance, such as the actual plasma membrane. Now we have types of membrane channels, because our ions are not going to just pass easily through the membrane. Sodium and potassium are the main ions we're going to be discussing. We have leakage channels that leak, always opened. Potassium and sodium can move around. However, you should take a note that sodium is much more difficult to move across the membrane than potassium is. Okay, now we have gated channels. Some are chemically gated, also ligand gated, and these open with neurotransmitters. Voltage gated open in response to an electrical charge and action potential. And then mechanically gated are going to open or close in response to the changes to the actual receptors that was caused by these chemical or voltage gated channels. Now, when gated channels are open, ions are going to diffuse across the electrochemical gradients, across the concentration gradients, and towards opposite charge. And ion flow along the plasma membrane creates an electrical current, also known as your action potential. Now, the potential difference is when you have a potential to have a charge, a flow of electricity. From the cytoplasmic side to inside of the cell, the potential difference is approximately -70 millivolts. And it's made up by differences of intracellular fluid and extracellular fluid, as well as permeability of the plasma membrane. Now, inside the cell, the intracellular fluid has a low concentration of sodium, but a high concentration of negatively charged proteins or anions. Since the membrane is impermeable to these large anions, it's going to stay somewhat negative inside. Now, not much permeability to sodium. Potassium just flows along either direction. So we end up at rest with a negative interior of the cell. And to keep that cell in homeostatic condition, we have a sodium-potassium pump. It essentially sits there all day, picks up sodiums, throws them out of the cell, picks up potassiums, and puts them back in the cell. So ultimately, at rest, you have more sodium on the outside of the cell, which is positive. Sodium is positively charged. A sodium ion is positively charged. So it's the cell at rest is more positive than it will be once the sodiums are moved. Okay, types of potentials. Graded potentials are short-term, short-distance potential. Electrical currents potentials. Action potentials go down the entire length of an axon. Depolarization is reduction in membrane potential. So essentially, the outside of the cell becomes the outside of the cell becomes more negative, whereas the inside is increasing in positivity. Hyperpolarization is when you have a cell that membrane potential is moved away from zero, so it's even greater, so it decreases the chances of you having a nerve impulse. Gradient potentials are short-lived, localized changes in potential. And you see here a graded potential. You had a little bit of a stimulus, a neurotransmitter. It moved one little piece of potassium in. It caused a little small electrical wave there. Okay, now once you get enough neurotransmitter in the synapse, you get a whole bunch of graded potentials. And a lot of, initially, you have enough ligand-gated channels. So a lot of sodium is moved inside. At that point, that electricity caused by the sodiums moving in start to open voltage-gated sodium channels, which basically all along the line of the axon. And so I put dominoes here. Once you get an action potential started, the negative charge from a sodium going down triggers the negative charge to push this one down, and then to push this one down, and so on and so forth, until all these dominoes fall down, or all the sodiums go in, and you get a wave of negative charge along the membrane. So the action potential is long distance electrical communication, and it does not decrease in magnitude. Once it starts, it just completely goes like rolling a ball down a hill. At resting state, only sodium potassium channels, all sodium potassium channels are closed. Only the leakage channels are open. I should have said gated channels are opened. Now, each sodium channel has 2 voltage-sensitive channels. These are your gated channels. Activation channels that open when you activate them, depolarize. Inactivation channels, they close when you're through using them. They close at rest. Now, each potassium-gated channel has 1 voltage-sensitive channel. They're closed at rest, but after depolarization, they start to slowly open. And the depolarizing local currents open the sodium channels, and you basically, as I said with the dominoes, you get sodium moving in, until you get a whole wave of negativity along the nerve, along the axon. Okay, now when it is through with this action potential, the first thing that happens is the potassium gates opened. Potassium exits. Potassium is very easy to move across the cell membrane, and the cell becomes repolarized. But you can't get another action potential until you get your sodiums and potassiums reorganized. Hyperpolarization is when you have so much potassium on the outside, it's hard to get an action potential going. Okay, repolarization only restores the electrical condition. That's your potassium. So to get the ionic redistribution, which has to happen before you get another action potential, the sodium-potassium pump starts to move sodium out of the cell, puts potassium back in the cell until you get back to a resting homeostatic state. Sodium influx opens the original channels, and repolarization follows a wave of depolarization. And this shows the same thing we talked about. Sodium is going into the cells, and once you get enough of the local sodiums going in, you have a negative current that causes another sodium channel to be opened, a voltage-gated sodium channel to be opened, which triggers that to go in, and that negativity triggers the next sodium to go in until you have an entire negative outer cell. That's called our action potential. And because of the nature of it taking a little while to put your sodiums back to where they go in the first place, you have only a one direction, all or nothing event. You have an absolute refractory period because it takes a while to set your dominoes back up. It takes a little few seconds to put your sodiums back where they go. And this ends this section of the lecture, and thank you for listening.